Hello class, this section is on muscles and it goes over chapters 10, 11, and 12 in the textbook. 10 and 11 chapters are really just chapters on naming the muscles, which you should go over in lab. And this will kind of just be a general introduction over muscular tissue. So there's three types of muscles in the body. There's skeletal, cardiac, and smooth muscle. And there are over 700 skeletal muscles together. Together they form the muscular system. Here are the four unique characteristics of muscle tissue. They're excitable, meaning they can respond to stimuli. They have the ability to contract or shorten their muscle fibers, and that allows them to move. They're elastic, meaning it can return to its resting length when applied tension is removed. And they're also extensible, meaning they have the ability to stretch beyond their resting length. So here are the functions of skeletal muscle tissue. They help with body movement. They help to maintain your posture. We all probably have terrible posture right now because we're all sitting over our computer. They help to maintain temperature regulation. That means your muscles will shiver or move to try to increase your body temperature. They help to store and move materials, especially through your digestive tract and your intestines. And they also help in supporting your body. The gross anatomy of skeletal muscle, each muscle is comprised of muscle fibers organized into bundles, which we called fascicles. The muscle fibers contain myofibrils, and myofibrils are composed of myofilaments. And this is just a great picture really explaining the structural organization of skeletal muscle. You can see here's a whole muscle here. Most likely this is the biceps brachii. And here are all of the fascicles that you can see inside a whole muscle. And if we remove a fascicle out, which is seen here, we can see how a fascicle is made up of muscle fibers, which are all these bundles. And if we remove out a muscle fiber, we can see how the muscle fiber is made up of myofibrils. And these are the myofibrils seen there. So this is just a great slide describing the levels of organization in a skeletal muscle. If we move down to myofibrils, you can see how myofibrils, which was the smallest unit we ended with in the last slide, is made up of myofilaments. And these myofilaments are thin and thick protein filaments, actin and myosin. Each muscle has 3 layers of concentric connective tissue composed of collagen and elastic fibers. And this connective tissue provides protection, sites for blood vessel and nerve distribution, and a means of attaching the muscle to the skeleton. The endomysium is the innermost layer, and the endomysium will surround and electrically insulate each muscle fiber, and that can be seen here. The perimysium surrounds each fascicles, and you can see the perimysium, the perimysium can be seen right there, and then the epimysium surrounds the entire muscle. Muscle attachments, at the ends of each muscle, all of the connective tissues merge to form a tendon, which will attach the muscle to a bone, skin, or another muscle. And a tendon is usually a cord-like in appearance, as is shown here, but sometimes it can kind of spread out and become a flat sheet, which we call an aponeurosis. Most muscles extend over a joint and have attachments to both articulating parts of the joint. The less movable point of the attachment is called the origin, and that's usually more proximally located to the trunk. And the more movable point of attachment is called the insertion, and the insertion will be attached to the part of the body that actually will move. Tendonitis, whenever you see this suffix itis on the end of a word, that always means inflammation. So tendonitis is inflammation of a tendon, and it's usually due to overuse of a specific tendon and muscle over time, usually to some sort of muscular imbalance. It's really common in the rotator cuff. Due to tendon injuries, and tennis elbow is known as lateral epicondylitis, and it involves inflammation of the extensor tendons in your forearm, and that can be seen here. Also, you'll get tendonitis with muscles of the rotator cuff. The microscopic anatomy of skeletal muscles, there's many components of a skeletal muscle fiber that are typical of a common cell, but they're named differently. So for example, the sarcolemma would be similar to the plasma membrane. I should point out right here that a skeletal muscle cell or a muscle fiber is the same as a muscle cell. So the sarcolemma, which surrounds it, would be the same as a plasma membrane on a typical cell. The sarcoplasm would be similar to the cytoplasm of a cell, and the sarcoplasmic reticulum, which will hold calcium ions, is similar to smooth ER in a typical cell. Two unique structures that are unique to muscle fibers themselves are transverse or T-tubules, which are deep invaginations of the sarcolemma. They extend into the sarcoplasm, and they just form a network that nerve impulses can travel and spread quickly throughout the muscle fiber. Also unique to muscle fibers are terminal cisternae, which are just blind sacs of that sarcoplasmic reticulum. Myofibrils then are cylindrical structures that extend the entire length of the muscle fiber cell. They have the ability to shorten, and this is what will result in a contraction of the muscle fiber. Then myofibrils are made of myofilaments, and these are just short bundles of protein. They do not run the entire length of the muscle fiber, but they're organized into repetitive groupings that we'll look at. And they can be thick filaments or thin filaments. Thick filaments are myofilaments that are composed of bundled molecules of myosin. They are twice as thick as thin filaments. They have an elongated tail and a head at the end. They kind of look like a golf club. And the heads of the golf club will form cross bridges or connection pieces to the thin filaments during contraction. The thin filaments then will be much smaller in diameter. They are composed of two strands of kind of the spherical molecules twisted around each other. And there are two regulatory proteins that are also part of the thin filament. Tropomyosin is this kind of long spaghetti-like orange structure. Tropomyosin covers the myosin head binding sites, and troponin, or the troponin complex, is a protein that's attached to the thin filaments. So myofilaments and how they band together, the thick and thin filaments kind of overlap each other, kind of as if you were to put your fingers together with them overlapping. And you'll see here the thin and thick filaments overlap each other. One sarcomere, a sarcomere is known as the contractile unit of muscle shortening or contraction, and they describe it as one Z disc to the next. And the Z disc are the parts of the myofibril that are connected to the ends of the thick and the thin filaments, excuse me. And what happens is during contraction, the sarcomere will shorten and the thick and thin filaments will slide past each other. So here's the organization. You have dark bands, which are called A bands. They contain the entire myosin molecule and some overlapping actin. And then the light bands, I bands, contain only thin filaments. Using an electron microscope, you can actually see this under a microscope, which is actually fun to see. I'll let you guys read through the descriptions of the other bands and zones. The M line is a protein meshwork in the H zone that keeps the thick filaments aligned, and the Z disc is the protein structure in the middle of the I band that serves as the attachment for the thin filaments. So here is the sarcomere then. It's the functional contractile unit in a skeletal muscle, and again it's defined by one Z disc to the next Z disc. And myofibrils will contain multiple and repeating sarcomeres, and again each sarcomere will shorten as the muscle fiber contracts and your thick and thin filaments slide past each other. So contraction of skeletal muscle fibers, this is known as the sliding filament theory. Muscle fibers will shorten, and this generates tension in the muscle. The sliding filament theory you guys will cover much more in depth in physiology, but we want to give you a little basis for it. During contraction, your thick and thin filaments will interact or grab hold of each other and form a cross bridge. And this causes the following changes within the sarcomere that I'll let you read about those zones. The length of your thick and thin filaments will never change, whether the muscle is contracted or relaxed, only their position changes as they slide past each other. And this is a great picture showing a relaxed biceps brachii and showing a contracted biceps brachii. And you can see how the sarcomere has shortened and there's more overlap between the thick and thin filaments. These are the events of contraction. There's a cross bridge formation where the myosin head will attach to the actin filament. And then there's a power stroke, which is where the myosin head will pivot and pull the actin filament toward the H zone. The cross bridge will then detach due to the presence of ATP or energy. And then the myosin head will cock back into position to get ready to attach again. So this takes you through the steps of contraction, and I would encourage you to pause the PowerPoint slide at this point and read through them. What's basically happening here is just the myosin head is attaching to the actin and pulling the actin to slide past it so the sarcomere can shorten. One thing to note that is important, you need ATP for the myosin head to detach and to get back into position. In death, right when a body will die, and ATP is no longer available, muscles might stay attached, so the myosin head might stay attached to actin, and this causes muscle stiffening called rigor mortis, and this is why in death, several hours after death, the muscles still might be stiff because there's no longer ATP present to release the myosin head from the actin. Eventually the proteins will degrade and the muscles will really relax, but that's called rigor mortis. In order to contract, a skeletal muscle must be stimulated by a nerve ending. So nerves are directly attached to your skeletal muscles. They'll propagate an electrical current, which will travel along the sarcolemma of the muscle fiber and basically release calcium for the sarcoplasmic reticulum. And calcium is what is needed for what we call this excitation, contraction, and coupling of the skeletal muscle contraction. Skeletal muscles will be stimulated by motor neurons. Axons of these neurons travel into the nerves to the muscle cells. They will branch profusely or into many pieces as they enter muscles, so that each axonal branch of that nerve will form what we call a neuromuscular junction with a single muscle fiber. And this is really interesting to see. So each branching from a motor nerve will make sure that each branch of that nerve will travel to each muscle fiber. And at this neuromuscular junction is where the motor neuron will come into close proximity to that muscle fiber. Components of the neuromuscular junction, there's a synaptic knob, which is the expanded tip of that axon nerve branch. Synaptic vesicles will be filled with acetylcholine, which is the neurotransmitter that'll be released, that will travel through the motor end plate and the synaptic cleft, and they will bind to the acetylcholine receptors. Acetylcholine esterase will just be an enzyme that rapidly breaks down acetylcholine to kind of stop that impulse from happening. So the nerve impulse will reach the terminal bulb. This will cause acetylcholine in the synaptic cleft to be released via exocytosis, and then acetylcholine will bind to receptors on the sarcolemma of the muscle fiber, and this will initiate what we call an action potential in the muscle. The action potential is what we call a depolarization event. This just means a polarity is changed, so there's a difference in charge moving across the fiber of that muscle cell, and that action potential will propagate into the membrane and eventually down to the T tubules to release calcium. So what does acetylcholine do? It will bind to its receptors at the motor end plate. The binding of it will open what we call chemically ligand-gated channels, and this causes sodium and potassium to diffuse out, and the interior of the sarcolemma becomes less negative. When something becomes less negative, this is called depolarization, and this will start the action potential to travel. So electrical conditions of a polarized sarcolemma. The extracellular face will be positive while the inside face is negative, and the difference in charge is what we call the resting membrane potential. The predominant extracellular ion is sodium, and the predominant intracellular ion is potassium. The sarcolemma is relatively impermeable to both ions, but an axonal terminal of motor neuron will release acetylcholine and will cause a patch of the sarcolemma to become permeable. That means sodium channels will open. Sodium, which is positive, will enter the cell and the resting potential will decrease and depolarization will occur. If a stimulus is strong enough, an action potential will be generated and will travel down that muscle fiber. Polarity reversal of the initial patch. This will change when voltage regulated sodium channels will now open in the adjacent patch, causing it to depolarize. And this will just spread along this depolarization wave down the fiber. So thus, the action potential will travel rapidly along the sarcolemma. Once initiated, it's important to remember that the action potential will be unable to be stopped and ultimately will result in the contraction of a muscle. Immediately after depolarization wave passes, the sarcolemma permeability will change. So the sodium channels will close, channels will open, and potassium will diffuse out of the cell, which will restore the electrical polarity, bringing the fiber back to normal. And that's what we call repolarization. The ionic concentration of the resting state is restored by the sodium-potassium pump. Excitation contraction coupling, once generated, the action potential will travel down the T tubules and will trigger calcium release from the terminal cisternae. Calcium is important because it will bind to troponin, which is a protein on actin. What does troponin do? Troponin, it'll cause the blocking action of tropomyosin to cease. Do you remember the tropomyosin was that piece of orange spaghetti? Troponin will cause a change in the tropomyosin, so the tropomyosin will actually move off the actin active binding sites, and they will now be exposed so that myosin can form a cross bridge or an attachment to the actin. The thin filaments will move towards the center of the sarcomere as the myosin pulls them in the power stroke, and then hydrolysis of ATP will power this cycling process. Calcium will be then subsequently removed back into the sarcoplasmic reticulum where it's stored. Tropomyosin will go back over its sites to block the myosin heads from attaching, and the muscle fiber will relax. So this is a great view of excitation contractions coupling, and I would just encourage you to pause the video at this point and read through this, but it shows how the action potential travels through the T-tubule, releases calcium from the sarcoplasmic reticulum. Calcium binds to troponin and causes the tropomyosin, which are these spaghetti-like orange structures, to come off the binding sites so that the myosin head can bind to actin. Slide the actin through. Extra calcium is removed by active transport back into the sarcoplasmic reticulum so that the muscle doesn't stay contracted forever, and the tropomyosin blockage is restored. A motor unit consists of a single motor neuron and the muscle fiber it controls. A motor unit typically controls only one or some of the muscle fibers in an entire muscle, and the size of the motor unit and degree of the control is inversely related. So what this means is that small motor units provide very precise control, and large motor units will provide not as specific or precise control. So you'll have a lot of small motor units in your fingers and in the muscles of your eyes, and not as many in the muscles of the back or the neck. Each muscle fiber obeys what we call the all or none principle, which means a muscle will contract completely or not at all. And when a motor unit is stimulated, all muscle fibers under its control will contract. Muscle tone refers to the constant tension in a resting muscle where motor units will be stimulated randomly to avoid fatigue. There's two types of muscle contraction, isometric or isotonic. Isometric is where the length of the muscle is constant, but the tension on the muscle is changing. And an isotonic contraction is where the tension is constant, but the length is changing. So a great example of this, of an isometric contraction, is you're holding yourself up in a plank. but you're not actually pushing the floor away. And an isotonic muscle contraction, I believe that's Arnold Schwarzenegger, but I'm not sure, is when you're actually doing a movement. Another great example of an isometric contraction is pushing with all your might against a wall, but you'll never be able to move that wall. So isometric, the tension is changing. You're putting tension on the muscle, but the length of the muscle isn't changing because you're not getting anywhere. In isotonic contraction, you'll put a constant tension on the muscle, and the muscle length is changing because you're able to lift the weight. And that is the end of this PowerPoint. Thanks for listening, guys.